Alright, let's go. Number 10. The Cheddar Man Pigmentation Surprise. In 1903, workers exploring Gough's Cave in Cheddar Gorge, England, pulled out the most complete Mesolithic skeleton ever found in Britain, roughly 10,000 years old. For over a century, he sat in a museum case as a face nobody could truly reconstruct. Scientists assumed the obvious. Ancient Britain, ancestor of modern Europeans, pale skin adapted to weak northern sunlight, the kind of assumption nobody thought to question. In 2018, researchers at the Natural History Museum in University College London, led by geneticist Selena Brace, sequenced DNA from bone powder taken from his inner ear. The result did not match the assumption. Cheddar Man carried the genetic markers for dark to black skin pigmentation and blue eyes, a combination modern viewers found almost impossible to picture. The team estimated skin pigmentation using established SNPs in a study whose full population genomics results were published in 2019 in Nature, Ecology, and Evolution. 3 explanations followed. Contamination, ruled out after repeated extraction and cross-referencing with other Mesolithic remains across Europe showing the same pattern. Recent admixture, ruled out. The sample predates the population movements that later lightened European skin by thousands of years. A statistical artifact of pigmentation prediction models. The most serious objection. And one Brace's team addressed by using multiple independent SNP panels that converged on the same result anyway. The lightening of European skin, it turns out, was not ancient. It was recent, patchy, and still finishing when farming reached the island. Later sequencing of other Mesolithic remains across Spain, Luxembourg, and Hungary showed the same dark skin, light eye combination, which is what finally closed the artifact objection down for most of the field. If Cheddar Man is representative, then the ancient European so many people picture, pale and blue-eyed, may not have existed for most of European prehistory at all. The face on your ancestry chart might be a modern invention. The past does not get lighter with distance. Sometimes it gets darker. Number 9. Homo Naledi's impossible burial behavior. In 2013, cavers exploring the Rising Star cave system outside Johannesburg radioed paleoanthropologist Lee Berger about a chamber packed with bones, accessible only through a chute barely 25 centimeters wide. Berger assembled a team of scientists small enough to fit. They were not expecting a new species, they were expecting a single accidental death. The standard model of human evolution ties symbolic behavior, deliberate burial, ritual, meaning-making, to large brains. Homo naledi had a brain roughly a third the size of a modern human's, comparable to a chimpanzee's. By that model, deliberate burial should have been beyond its cognitive reach. What Berger's team found inside the Naledi chamber were the remains of at least 15 individuals, arranged in ways that resisted simple explanation, alongside what the team later described as engraved geometric patterns on the cave walls. In 2023, Berger, Agustin Fuentes, and colleagues released a set of preprints through eLife arguing for deliberate burial and deliberate mark-making, dating the chamber activity to between 236,000 and 335,000 years ago. The failure parade started immediately. Water transport unlikely given the chamber's inaccessible geometry and the bone's preservation pattern. Predator accumulation, no tooth marks, no scattering consistent with carnivore activity. Accidental single-entry deaths. Contradicted by multiple individuals of different ages found in distinct, separated locations within the same deep chamber system. Peer reviewers pushed back hard on the burial claim specifically, with critics like Aurore Val and Mara Martin Torres arguing the sediment evidence does not support intentional graves. The debate remains unresolved in the literature. If a small-brained hominin carried its dead half a kilometer into total darkness for reasons we cannot reconstruct, then the line separating them from us was never about brain size at all. Nobody agrees on what those hands were doing in the dark. Nobody can ask them. Number 8. The Out of Africa Timeline Revision. In the 1960s, French and Moroccan researchers began excavating a barite mine at Jebel Irhoud, initially searching for evidence of Neanderthal populations known to have occupied North Africa. What they expected was a peripheral Neanderthal outpost. The standard model, built through the 20th century, placed the emergence of anatomically modern Homo sapiens in East Africa roughly 200,000 years ago, a tidy single-region origin point. In 2017, a team led by paleoanthropologist Jean-Jacques Hublin at the Max Planck Institute for Evolutionary Anthropology re-examined the Jebel-Irhoud remains using updated thermoluminescence dating on burned flint tools found alongside the bones. Published in Nature in 2017, Hublin's team dated the fossils to approximately 300,000 years old, pushing the emergence of Homo sapiens back by roughly 100,000 years, and placing it in North Africa rather than the East African cradle textbooks had settled on for decades. The failure parade for the old model followed fast. Misdating of earlier East African fossils as the definitive first undermined once Jebel-Irhoud's dates held up under repeated testing. A single point of origin, increasingly abandoned in favor of a pan-African model, where our species emerged gradually across multiple interconnected populations rather than one location. Morphological gatekeeping. The assumption that modern skulls must look fully modern immediately, undercut by Jebel-Irhoud's mixed features, a modern-looking face on an archaic, elongated brain case. There was no single Eden. There was no single morning our species began. Later fossil finds at Florisbad in South Africa and Omo Kibish in Ethiopia, both roughly contemporary with Jebel-Irhoud once redated, only reinforce the picture of a species assembling itself in pieces, across a continent, rather than stepping fully formed out of one valley. If your species doesn't have a birthplace, what exactly are you the descendant of? The map keeps growing new points of origin. None of them are final. Number 7. Neanderthal immune gene inheritance. In early 2020, geneticists Hugo Zeeberg and Svante Pääbo at the Max Planck Institute were not studying Neanderthals at all. They were combing through genome-wide association data trying to understand why COVID-19 severity varied so wildly between patients with similar health profiles. The standard assumption in modern genomics treats archaic human DNA, the roughly 2% of Neanderthal ancestry carried by non-African populations, as evolutionary background noise, mostly neutral, occasionally harmful, rarely relevant to a 21st century pandemic. Zeeberg and Pääbo found a cluster of genes on chromosome 3 that dramatically increased the risk of severe COVID-19 outcomes, and traced its origin directly to Neanderthal DNA inherited more than 50,000 years ago. Published in Nature in 2020, the study identified this Neanderthal-derived haplotype as the single strongest known genetic risk factor for severe COVID-19, carried by roughly 16% of Europeans and up to 50% of South Asian populations, and entirely absent in African populations with no Neanderthal admixture. The team ruled out coincidence. The haplotype block was too large and too precisely matched to Neanderthal reference genomes to be anything but inherited. They ruled out modern selection pressure creating a similar variant independently. The mutations were identical, not convergent. What they could not explain was why a gene variant carried through 50,000 years of relative genetic silence would suddenly become one of the deadliest inherited traits in a global pandemic nobody could have predicted. Somewhere in your genome, if your ancestry traces through Europe or Asia, an extinct species may still be deciding how sick you get. The people who vanished 40,000 years ago never fully left the room. Number 6. The unexplained Y-Chromosome bottleneck. In 2015, population geneticist Monica Carmen and a large international team were mapping global Y-chromosome diversity to refine the male mutation rate, a routine calibration exercise for the molecular clock used across human genetics. The standard model predicted a roughly steady decline in genetic diversity as populations grew and spread, mirroring the general shape of maternal mitochondrial DNA lineages over the same period. Carmen's team found something the mitochondrial data did not show: a severe, sudden crash in Y-chromosome diversity worldwide, concentrated between 4,000 and 8,000 years ago. Published in Genome Research in 2015, the study estimated that the effective number of reproducing males dropped to roughly 1/20th of the effective number of reproducing females during this window. a bottleneck with no equivalent crash on the maternal side. The failure parade followed. A general population crash ruled out since female lineages show no matching collapse in the same period. Disease selectively killing men, no pathogen candidate fits a bottleneck this synchronized across separate continents. The leading explanation, proposed by Carmen's team and expanded by later researchers, points to intensifying warfare between patrilineal kin groups during the early Bronze Age, where entire male lineages were wiped out in group versus group conflict while women were absorbed into the winning groups. It remains a hypothesis, not a settled fact, and no single archaeological site confirms it globally. A rival explanation, proposed by other researchers working the same dataset, points instead to the rise of patrilineal inheritance systems, where property and status pass through a shrinking number of male lines even without direct violence. Neither model has been ruled out, and both would need to have operated across separate continents at almost the same time. If the theory holds, roughly 19 out of 20 men alive during that window left no sons who survived to have sons of their own. Something erased entire bloodlines of men, everywhere, at once. Nobody has found the weapon. Number 5. The unknown ghost population admixture. In 2019, population geneticists were fine-tuning genome-wide diversity statistics for West African populations. A technical step meant to correct reference panels used across all of human genomics. The standard model held that all modern human genetic diversity could be traced back to known lineages, the deep African branches, plus the Neanderthal and Denisovan admixture already documented in populations outside Africa. Statistical geneticists Arun Durvasula and Sriram Sankararaman at UCLA found something the known lineages could not account for. Published in Science Advances in 2020, their analysis estimated that between 2 and 19% of the genomes of modern Yoruba and Mende populations in West Africa derive from an unidentified, deeply diverged archaic human lineage, one that split from the ancestors of modern humans roughly as long ago as the Neanderthal Denisovan split itself, but with no fossil ever recovered. The team ruled out contamination from known archaic genomes since the signal did not match Neanderthal or Denisovan sequence. They ruled out a statistical artifact of population structure, running the analysis across multiple independent methods that converged on the same estimate. What could not be ruled out? Because no bone or tooth or fragment has ever surfaced to test it directly, is the identity of the population itself. An entire branch of the human family tree, contributing meaningfully to living people today, with no face, no skeleton, no name. Geneticists call it, without irony, a ghost. Something interbred with our ancestors in Africa and left descendants everywhere except the fossil record. Number 4. The Toba catastrophe bottleneck. In the 1990s, anthropologist Stanley Ambrose at the University of Illinois was investigating why global human genetic diversity looked so surprisingly low compared to that of other great ape species that had existed far longer without expanding across the planet. The standard expectation was gradual accumulation, more time, more diversity, following the typical logic of population genetics. Ambrose connected the shockingly low diversity to a specific geological event, the eruption of the Toba supervolcano in Sumatra roughly 74,000 years ago, the largest volcanic event of the last 2 million years. Published in the Journal of Human Evolution in 1998, Ambrose's model proposed that Toba's eruption triggered a volcanic winter lasting years, collapsing global temperatures and crashing the human population to as few as 1,000 breeding pairs, a bottleneck severe enough to leave its signature in the genome of every person alive today. The failure parade has run for over 2 decades. Archaeological sites in India, below and above the Toba ash layer, show continuous stone tool traditions with no interruption, a direct challenge to a species-wide collapse. Climate models by later researchers suggest the volcanic winter may have been milder and shorter than Ambrose's original estimate. Some geneticists now argue the low diversity reflects a slow population expansion out of a small ancestral group, not a sudden catastrophe at all. The debate has never fully resolved either way, publication by publication, for over 25 years. Ice core data from Greenland does show a sulfate spike consistent with a major eruption around this period, which keeps the volcanic winter hypothesis alive even as the size of its human impact stays contested. If Ambrose was right, every person reading this exists because roughly 1,000 couples survived a volcanic winter that could easily have gone the other way. The nearest we came to not existing at all might be sitting under a layer of ash in Sumatra. Number 3. Mitochondrial Eve's statistical paradox. In 1987, biochemist Alan Wilson and his graduate students Rebecca Cann and Mark Stoneking at UC Berkeley were trying to build a molecular clock for human mitochondrial DNA, a technical project meant to date branch points in the human family tree. The standard assumption of the time was that multiple, geographically separate human populations had evolved into modern humans independently and roughly simultaneously across the globe, the multi-regional model. Cann, Stoneking, and Wilson's analysis, published in Nature in 1987, traced every mitochondrial lineage in a global sample of 147 people back to a single common maternal ancestor, living in Africa an estimated 200,000 years ago. The press nicknamed her Mitochondrial Eve, and the multi-regional model never fully recovered. The failure parade targeted the finding itself. Critics initially challenged the statistical tree-building method used, arguing the software could produce misleading tree shapes, a fair critique that later, more rigorous analyses with larger datasets nonetheless confirmed the core finding. Other critics pointed out, correctly, that Eve was not the first woman or the sole living woman of her time, simply the only one whose unbroken maternal line survives in every person alive today. Every other maternal line from her era ran out of daughters and went extinct. That distinction rarely survives the retelling. What remains genuinely unresolved is why the bottleneck down to a single surviving maternal line happened at all, in a population that was not, by any archaeological measure, undergoing a mass extinction event at the time. Every mother you have ever had, traced back far enough, converges on one woman in one place, for reasons nobody has fully explained. Every line but one went extinct. Nobody agrees on why hers didn't. Number 2. The Denisovan-Human Hybrid Fossil. In 2012, archaeologists at Denisova Cave in the Altai Mountains of Siberia had been slowly sifting through decades of accumulated bone fragments, hoping to identify anything worth full genomic sequencing among thousands of unremarkable splinters. The standard expectation, based on everything known at the time, was that interbreeding between archaic human species, Neanderthals and Denisovans, was rare enough that direct physical evidence of a single hybrid individual would likely never be found. Geneticist Vivian Slan and colleagues at the Max Planck Institute sequenced a small bone fragment catalogued as Denisova 11 and nicknamed Deni, and found something no lab had ever documented before. Published in Nature in 2018, Slan's team determined that Deni was the direct offspring of a Neanderthal mother and a Denisovan father, a first-generation hybrid, dated to roughly 90,000 years ago. The team ruled out contamination through repeated, independently replicated sequencing across multiple lab batches. They ruled out a Neanderthal Denisovan population with generations of prior mixing, since the genome showed exactly a 50-50 split, consistent with immediate first-generation parentage, not a blended lineage. What the discovery could not explain was how common this actually was. A single confirmed hybrid, found in one of the smallest, least statistically probable bone fragments in the collection, implies a rate of interbreeding between distinct human species that current models cannot yet quantify. Slan's team noted that Deni's Denisovan father also carried some Neanderthal ancestry from generations earlier, meaning at least 3 separate cross-species encounters converge in one individual's genome. If finding one hybrid this quickly was this improbable, how many more encounters between our closest extinct relatives are we simply not finding? One bone fragment rewrote how often extinct humans loved, or at least mated, across species lines. Number 1. The Denisovan ghost lineage. In 2008, archaeologists working Denisova Cave sent a single, unremarkable fingerbone fragment found in a layer alongside stone tools to the Max Planck Institute for what was expected to be routine confirmation. Another Neanderthal, another data point. The standard model at the time recognized exactly two human lineages in Eurasia during this period: modern humans and Neanderthals, with archaic hominin fossils elsewhere on the continent generally assumed to fall into one category or the other. In 2010, a team led by Svante Pääbo sequenced the mitochondrial and later the full nuclear genome from that fingerbone and found genetic sequences that matched neither modern humans nor Neanderthals. Published in Nature in 2010, Pääbo's team announced an entirely new archaic human lineage, the Denisovans, identified almost entirely from DNA, not morphology. An inversion of over a century of paleoanthropological method. To date, the entire physical record of Denisovans consists of a finger bone, a handful of teeth, and one partial jawbone found later in Tibet. No complete skull, no reconstructed face, no full skeleton, anywhere, ever. The failure parade here is not about disproving Denisovans. It is about the limits of ever fully knowing them. Attempts to reconstruct their appearance from DNA, predicted traits remain speculative, unconfirmed by any matching skull. Their geographic range, inferred almost entirely from the DNA they left behind in living Melanesian and Aboriginal Australian populations, up to 5% of some living genomes, vastly outpaces the tiny handful of physical sites where their bones have actually surfaced. An entire human species confirmed to have existed, confirmed to have interbred with our direct ancestors, and yet nobody, not one living scientist, knows what their face looked like. They are real enough to be inside you, and unknown enough that no one could pick them out of a crowd. 12 discoveries. One pattern. Every genome carries a species that no longer exists. A bottleneck nobody fully explains. A ghost with no face. This is not the story of ancient people who were simple, or primitive, or separate from us. It is the story of how much of what feels like you, your skin, your immune system, your survival at all, was decided by extinct strangers, in the dark, tens of thousands of years before anyone alive could ask them why. The DNA is not confused. It is precise, replicated, published, peer-reviewed. We are the ones still catching up to what it has been quietly telling us all along. If you want to see more videos like this, click the video on screen now.